Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs of the year 2015. If you are new to my channel, please do like, share and subscribe. Then coming to the year June 2015. In bacteria, heat shock response is primarily controlled by, so it is controlled by sigma 32. Answer is option 2. These are part B questions. Then, in type 2 splicing, a free 2' O of an internal adenosine makes a nucleophilic attack on the 5' P of the first base of an intron. Option 2 is the right answer. Then next one. Copying errors occurring during replication are corrected by the proofreading activity of DNA polymerases that recognize incorrect bases at the 3' end of the growing chain and remove them by 3', 5' exonuclease activity. Answer is option 3. Then, during each cycle of chain elongation in translation, how many conformational changes does the ribosome undergo that are coupled to GTP hydrolysis? So, it is 2. That is, answer is option 3. 3 is the answer. Coming to part C. When circular plasmids having a centromere sequence are transformed into yeast cells, they replicate and segregate in each cell division. However, if a linear chromosome is generated by cutting the plasmid at a single site with a restriction endonuclease, the plasmid are quickly lost from the yeast. It is known that genes on the plasmids are lost because of the instability of the chromosome ends. What could be done so as to restore its stability and be inherited? So you have to read this statement, that is instability of the chromosome ends. Concentrate on this statement, instability of the chromosome ends. That means it has got something to do with the telomere sequences. So if you see option 3, by incorporating telomere sequences to the ends of the plasmid. Option 3 is the right. Then next one. Eukaryotic DNA polymerase alpha has tightly associated primase activity but moderate processivity. DNA polymerase epsilon and delta are highly processive but lack primase activity. Given below are 4 statements about leading and lagging strand synthesis in eukaryotes. Which one is true? Both leading and lagging strand are synthesized by DNA polymerase alpha. Leading by epsilon and lagging by delta. Both are not synthesized by DNA polymerase alpha. So statement 1 is not correct. Entire leading and lagging strand are synthesized by delta and epsilon. Eukaryotic replication is primer independent process. It is primer dependent process. So statement 2 is also not correct. Only the lagging strand synthesis needs primer and synthesized by DNA polymerase alpha. Both leading and lagging strand, they require a primer. So option 3 is also not correct. Primers for both the strands are synthesized by DNA polymerase alpha followed by polymerase switching with epsilon and delta. That is correct. Then next one. In contrast to lac operon, if two bacterial strains P1 and P2 with the genotype OC I plus Z minus and O plus I minus Z plus respectively were used to produce mero-diploid daughter strain D, which of the following statement correctly predicts the expression of Z gene, that is beta galactosidase activity, in all the 3 strains? O plus, I plus and Z plus denote the wild type allele of the respective genes. So if you see this one, first case, that is P1, OC I plus Z minus, there won't be any expression of beta galactosidase because it is Z minus. Operator is constitutive, repressor is positive. It is not mutated, but operator is constitutive, so repressor cannot go and bind to the operator. But Z is minus, beta galactosidase is not produced. So P1 no expression. Then next one, O plus I minus Z plus. It is I minus, repressor is mutated. So it will not bind to the operator. So Z plus is there. So it will be constitutive production of Z plus. So P2 is constitutive expression. Now coming to the mirror diploid strain, that is D. OC I plus Z minus, O plus I minus Z plus. So I plus is there. So here it is in O plus I minus, it is I minus, whereas OC it is I plus. So I plus is a transacting element. The repressor protein is a transacting element. So the repressor protein can bind to the operator, this operator it can bind, but not to operator C. It can bind to the O plus operator and prevent the expression of lac Z gene. So D is inducible expression. So P1 is no expression, P2 is constitutive expression, D is inducible expression. And the answer is option 1. The next one. In order to study the transcription factor TF2H, it was cloned from a large number of human subjects. Surprisingly, the subjects having mutation in the TF2H also showed defects in their DNA repair system. Given below are the explanation. Now, TF2H mutated individuals showed defects in DNA repair system. That means TF2H is involved in DNA damage repair. DNA damage is always associated with transcription inhibition. That is not correct. TF2H has no role in DNA repair. It has a role in DNA damage repair. In mammalian system, TF2H plays an active role in transcription coupled DNA repair process. That is true. Because of mutation in TF2H, transcription initiation is inhibited and incompletely synthesized mRNAs remain attached to the template DNA, leading to DNA damage. That is not correct. So the option is option 2, that is C only. Then next one. The end of most eukaryotic mRNAs is defined by the addition of a poly-A tail, a processing reaction called polyadenylation. The addition of poly-A tail is carried out by the enzyme poly-A polymerase. Given below are few statements about this process. Poly-A polymerase is template independent enzyme. That is correct. Poly-A polymerase catalyzes the addition of AMP from DATP. It is not from DATP, it is from ATP. So B is wrong. Poly-A polymerase is an RNA template dependent enzyme. That is also not correct. Poly-A polymerase catalyzes the addition of ADP. It is not ADP, it is AMP from ATP. So D is also not correct. Poly-A polymerase catalyzes the addition of AMP from ATP to the 3' end of mRNA. That is correct. Poly-A polymerase catalyzes addition of AMP from DADP. That is also not correct. So A and E are correct. Answer here is option 3. Then next one. With an intention to identify the genes expressed in an organism at specific stages of development, mRNAs were isolated from the given organism, cDNAs were synthesized, cloned in a suitable vector, and sequenced. A few of the cDNA sequences showed no matches with the genomic DNA sequence. Further, it was observed that these sequences were U-rich and found to be in stretches dispersed along the sequence. The following may be the possible reason for appearance of such RNA. So, which of the following is the most appropriate reason? Now they have found these sequences to be U-rich, so it must be something to do with guide RNA, because guide RNA either inserts or deletes U. So splicing cannot be the answer, alternate splicing cannot be the answer, trans-splicing no, guide RNA mediated introduction of U's involving endonuclease, terminal U transferase and RNA ligase. D is correct. Deaminations converting C to U. That is also not possible. So the answer here is option D only. Then next one. When one isolates ribosomes from bacterial lysate apart from 70S, 50S and 30S ribosomal subunit, one also finds small population of 100S, 130S, 150S subunits. EDTA dissociates these large ribosomal subunits into 50S and 30S, suggesting that they have both the subunits. Upon addition of cations, they reassociate into 70S, but none of the other forms could be detected. What is the reason for not obtaining greater than 70S forms? Now EDTA, it is a divalent chelator and it chelates Mg2 positive ions. Okay. Now coming to the statements. The effect of EDTA cannot be reversed by the addition of cation. They have already mentioned in the question that it can be reversed. Upon addition of cations, they reassociate into 70S. So first statement is not correct. 100S, 130S, 150S are modified form of ribosome that are irreversibly damaged by EDTA. It is not irreversibly damaged, it is reversibly damaged by EDTA. 100S, 130S, etc. represent polysomes that cannot be reassembled de novo without other cellular components. That is correct. That is why upon addition of cations, they reassociate into 70S forms and not to 100S, 130S and 150S subunits, because they are polysomes. They are obtained as an experimental artifact. That is also not correct. Coming to the year December 2015. Coming to first question, these are part B questions. A cell line deficient in salvage pathway for nucleotide biosynthesis was fed with medium containing N15 labeled amino acids. Purines were then extracted. Treatment with which one of the following amino acids is likely to produce N15 labeled purines? So it is glutamine. Answer is option 3. Then next one. An antibiotic that resembles the 3' end of charged tRNA molecule is, it is puromycin. Next one. Alpha-amanitin is a fungal toxin which inhibits eukaryotic RNA polymerases. The 3 eukaryotic RNA polymerases show differential sensitivity to the toxin. Which one of the following order, higher to lower, is the correct in respect of sensitivity towards alpha-amanitin? So it is RNA polymerase 2, because amanitin inhibits RNA polymerase 2 more than RNA polymerase 3 and RNA polymerase 1. So it should be RNA polymerase 2 greater than RNA polymerase 3 greater than RNA polymerase 1. So the answer here is option 2. In eukaryotic replication, helicase loading occur at all replicators during the G1 phase. Coming to part C questions. Which of the following statement correctly applies to proteins which are translated on the rough endoplasmic reticulum? So from endoplasmic reticulum, the proteins will be transferred to Golgi, from there to lysosomes, then to plasma membrane and then cell exterior. So the answer here will be option 2, that is protein targeted to lysosomes, plasma membrane and cell exterior. Answer is option 2. A culture medium contains two carbon sources. One is preferred carbon source glucose. The second one is non-preferred sucrose, that is lactose. Which one of the above, which one below is correct regarding the nature of the growth curve of E. coli cultured in this medium? So a culture medium, it contains two carbon sources. One is glucose, that is preferred, and the other is lactose. So which one of, so it will show a dioxic growth curve. E. coli will show a dioxic growth curve. So it will have a lag phase will be observed between two exponential phases. Answer is option 3. Then next one. You have labeled DNA in a bacterium by growing cells in a medium containing either N14 nitrogen or a heavier isotope N15. Furthermore, you have isolated pure DNA from each organism and subjected to cesium chloride gradient density centrifugation, leading to their separation of light and heavy forms of DNA to different locations in the centrifuge tube. In the next experiment, bacteria were grown first in a medium containing N15 so that all the DNA made by cells will be in the heavy form. Then these cells were transferred to medium containing only N14 and allowed the cells to divide for one generation. DNAs were extracted and centrifuged as above in the cesium chloride gradient. A hybrid DNA band was observed at a position located between and equidistant from the N15-14 DNA bands. Based on the above observation, which one of the following conclusion is correct? So what they have done here is they have explained the whole experiment, which proves that DNA replication is semi-conservative. So the answer here is replication is semi-conservative. Answer is option 2. Then next one. Although ribonucleoside triphosphates are present at approximately 10-fold higher concentration than deoxyribonucleoside triphosphates in the cell, but they are incorporated into DNA at a rate that is more than 1000-fold lower than DNTPs. This is because DNA polymerase efficient' discriminates between RNTPs and DNTPs because its nucleotide binding pocket cannot accommodate a 2' OH on the incoming nucleotide. 2' OH will be present in RNTPs, but it will not be present in the DNTPs. So the answer here is option 3. Then next one. The mismatch repair activity of E. coli requires in misincorporated bases which is not removed by the proofreading activity of DNA polymerase. However, while doing so, it has to decide which strand of the DNA is newly synthesized and which one is parental. Mismatch repair system does it by which one of the following ways? So here they have asked about mismatch repair mechanism. So it recognizes the hemimethylated GATC sequence nearby. Answer is option 4. And A, adenosine will be methylated in GATC and parental strand will be methylated but the daughter strand will be non-methylated. Answer is option 4. Then next one. Enlisted below are different types of RNAs produced in the cell and their functions but not in the same order. Choose the correct combination. So snRNAs, they function in a variety of processes including splicing of pre-mRNA. A3. siRNAs, they turn off gene expression by directing degradation of selective mRNAs. C. miRNAs regulate gene expression by blocking translation of selective mRNAs. C2. snRNAs used to process and chemically modify rRNAs. D4. So the answer here is option 2. A3, B1, C2 and D4. Then next one. In prokaryotes, the initiator tRNA is first charged with methionine, followed by the addition of a formyl group to the methionine by the enzyme MET tRNA transformylase. Given below are several statements in this context. Which of the following is true? All prokaryotic proteins have formyl methionine at their amino terminal end. No. Then deformylase removes the formyl group from the amino terminal methionine. That is correct. All prokaryotic proteins have methionine at their amino terminal end. That is also not correct because aminopeptidases often remove the amino terminal methionine. Answer is option B and D, that is option 4 is the right answer.